Good morning. Today we are going to continue with autotrophic nutrition. Now, as discussed in the previous class, nutrition is of two types, autotrophic and heterotrophic. Auto, self, hetero, different. That is, when the organism manufactures its own food, which is seen in green plants, certain bacteria, that is referred to as autotrophic nutrition. Heterotrophic we'll be dealing with later. Today we'll continue with autotrophic nutrition. Now, in case of autotrophic nutrition, energy and carbon requirements are fulfilled by the process known by the name of photosynthesis. Now, as explained previously also, if we know the meaning of different terms that make up a single term, we will be able to find out the definition, we will be able to make the definition when we are writing in our exams. We don't need to memorize anything. Now here, the word term photosynthesis itself is made up of two terms, photo and synthesis. Photo meaning light, and synthesis, that is making or manufacture. Now making and manufacture of what? Of food and oxygen. Now the raw materials which are required for photosynthesis are carbon dioxide and water. Overall, 6 molecules of carbon dioxide react with 12 molecules of water to make up a molecule of glucose and 6 molecules of water. The energy conversion that is involved in photosynthesis is light energy getting converted to chemical energy. This light energy is obtained from the sun. And the chemical energy is stored in the form of bonds of carbon compounds, basically glucose. In case of plants, since plants keep on making more and more glucose, it becomes too much for the plant to utilize it immediately, and therefore that glucose gets converted to starch and it gets stored in the plant. This glucose is used to make up the different parts of the plant, whether it is a new leaf, a new rootlet, and in future flowers and fruits. Also, this starch is stored in certain plants like potatoes as a part which can be consumed by animals. And this reserve food material is present not only in plants, it's also present in animals. In case of humans, just like starch is the stored form of carbohydrate, in humans, the stored form of carbohydrate is glycogen. Now, there are 3 basic steps or events that occur during photosynthesis. The first event is absorption of light. Now this absorption of light is carried out by the green pigment chlorophyll, which is present in the chloroplast of the cells of the leaf. The second step is conversion of this light energy, which is absorbed by chlorophyll, into electrical energy. So light energy gets converted to electrical energy. Now just now I said a few minutes ago that the conversion is light energy to chemical energy. But here I am saying the second step is, or rather the event during photosynthesis is conversion of light energy to electrical energy. Why? Because this electrical energy will then be used to split the molecule of water. This splitting of water leads to formation of hydrogen and oxygen, and it is this oxygen which is released in the end as a byproduct of photosynthesis. So all the oxygen that we obtain at the end of photosynthesis is obtained from the hydrogen, that is the result of a splitting of water through electrical energy, which was converted from solar energy. This splitting of water has resulted in formation of two elements, that is hydrogen and oxygen. This hydrogen, the third step is reduction of carbon dioxide. The hydrogen which is obtained as a result of a splitting of water is then used to reduce carbon dioxide into a carbohydrate. The first carbohydrate that is formed is CH2O. 6 such steps are repeated, which result in the formation of a molecule of glucose C6H12O6. Now, whether the stored form that is produced during photosynthesis, the reserved food, that is starch, is an indication of photosynthesis has occurred. So whether photosynthesis has occurred or not, we can check through very simple starch test. Now what exactly is a starch test? A starch test is a test in which we check whether starch is being produced, it is present in the part of the plant or not. The different steps of a starch test are: first, boiling the part of the plant, boiling of leaf. Why leaf? Because it's basically the leaf which is involved in the process of photosynthesis. First we boil the leaf. Now why do we boil the leaf? We boil the leaf so that we stop all the reactions occurring within the leaf. Stopping of reaction is required so that no new substance is formed, so that exactly whatever substances are present remain as such. We do not want any addition or deletion of substances from the leaf. And moreover, even after detaching the leaf from the main plant, the leaf continues to live. It continues to live for a very long time until and unless desiccation, that is drying up, settles in, and then only the leaf dies. So we have to first kill the leaf, so we boil it. Then after boiling the leaf in water, we then boil it in alcohol. Why in alcohol? Because alcohol decolorizes chlorophyll. The green color needs to be removed from the green part of the plant, so that if iodine will be added to carry out the starch test, we can get an exact color of iodine, because it's the color of iodine which indicates the presence or absence of a starch, but the green color will interfere in the formation of in carrying out of starch test. So we boil it in alcohol. After that we wash the leaf. Now why washing of leaf? Again, to remove alcohol. Why alcohol needs to be removed? So that again it doesn't react with iodine further. Now once the leaf has been boiled in water, then in alcohol, it has been washed. Now we put in a few drops of iodine on the leaf. Formation of blue-black iodine is an indication of presence of a starch. If the color of iodine remains as brownish, it indicates that a starch is absent. So this is the way we carry out the starch test. Now why we need to carry out a starch test when we are dealing with photosynthesis is because we need to be sure of what exactly are the requirements of photosynthesis. So we carry out different tests for requirements of photosynthesis. The first test that we rather the first substance that we check out whether it is required or not is chlorophyll. Whether chlorophyll is required or not, that we check. For that we use a variegated leaf. Now what are variegated leaves? Variegated leaves are those leaves like that of croton which have certain areas within them that are non-green. A non-green area indicates the absence of chlorophyll. If it's yellow or whitish part of the leaf, it indicates that chlorophyll is missing. Yellow indicates presence of xanthophyll, white indicates the presence of leucoplast but not chloroplast, which are present, which contain chlorophyll, which will be able to absorb sunlight. Now what we do with variegated leaf, we carry out the same starch test, boiling in water, boiling in alcohol, washing, and then adding in a few drops of iodine. When we carry out the starch test on a variegated leaf, we see that the green parts will turn blue-black and the non-green parts will remain as such, will retain the brown color of iodine, which shows that chlorophyll, wherever it is present, in whichever part of the leaf chlorophyll is present, there only photosynthesis has occurred. Otherwise, no photosynthesis. The second test that we need to carry out is whether light is required or not. So what do? We take a simple leaf and we cover a part of the leaf with a black paper and leave it under the sun. After some time, when we carry out the starch test, we see that the part on the whole leaf, after removing the paper, we carry out the starch test, and what we observe is that the part which was exposed to light has turned blue-black, but the part which was not exposed to light, that is the part which was covered by the black paper, doesn't have any starch. It remains brown as such, the color of iodine. Again, this indicates that light is required for photosynthesis. The third requirement we check is of carbon dioxide. Now, for carbon dioxide, what we do is we put a potted plant underneath a bell jar. We in fact take two potted plants, we put them both beneath bell jars. In one bell jar, we place potassium hydroxide, and in the inside the other bell jar, we do not place this potassium hydroxide. Now what this potassium hydroxide does is it absorbs the carbon dioxide of air. So when carbon dioxide is missing from bell jar, photosynthesis will not occur, and in such a case the leaves of plant, when a starch test will be conducted on them, will not show blue-black color, the color of iodine when starch is present. So again we can continue checking for water and other, but water is difficult because if we dehydrate the plant, then the plant will die, because plant does not require water only for photosynthesis, so it requires water for its existence also. It requires water for its various metabolic reactions going on within the plant body. So checking for water is difficult, but definitely we know without water life is not possible. So definitely photosynthesis will also be not possible. Now these are the requirements of photosynthesis: chlorophyll, light and carbon dioxide, without which photosynthesis will not occur. Now we come on to the part of the leaf which is responsible for taking in of carbon dioxide, because we know chlorophyll is present in the chloroplast in the cells of leaf. We know that carbon dioxide is present in the atmosphere, and we know light is all around the plant during the daytime. But how come this carbon dioxide is actually reaching inside the chloroplast for photosynthesis to occur? For that, carbon dioxide needs to be taken in, it needs to be diffused inside the leaves, and that taking in of carbon dioxide is done through small pores known by the name of stomata. The term stomata means opening or a pore. Now these stomata, which are present mainly on the undersurface of the leaf, or especially of dicots. Now if you look at dicot leaf, it is very glossy on one surface, on the other surface it is rough, the one which is beneath, downward. Now the one which is glossy is dark green in color, the one which is rough is light green in color. It is dark green, the above surface, the glossy surface is dark green because of presence of chloroplast. The downward surface is rough because of the presence of these structures known as stomata. Now, overall this is the structure of stomata, which also contain a large number of chloroplasts inside them. They are made up of, a stomata is made up of two bean-shaped cells known as the guard cells, and there is a pore in between, and it is this pore known as stoma, opening from where actually carbon dioxide gets inside and oxygen moves outside. This open stomata, these guard cells, they take in water, they become rigid, and the stoma opens, the pore opens, carbon dioxide is able to get in. During evening time, the guard cells, they lose the water, they become flaccid, the stoma closes, the exchange of carbon dioxide and water stops. In fact, it is this stoma which also helps to pull up water from the roots to the leaves through transpirational pull. Transpiration, the process of loss of water in the form of water vapors from the stomata. Because when the water is lost through stomata from the leaf, a low pressure risk of water is created within the leaf, which helps in taking up of water from the roots, or upliftment of water from the roots to the leaves. Now, these stoma, or rather stomata, also need to be closed when the exchange of gases is not required. In the daytime it remains open, carbon dioxide keeps on going in, oxygen keeps on coming out, water vapors keeps on coming out, but by evening this needs to be stopped. Excess carbon dioxide is not required by the plants in the evening or at night. So these stomata close. How do they close? Again, guard cells, they become flaccid, and that is due to loss of water from the guard cells. At night, now the question that should be coming in your mind is, then how come plants are able to breathe at night? At that time, the exchange of gases occurs through the surface of different parts of the plants which are living, direct diffusion from air to the cell of the plant and vice versa. But in daytime, when large amount of carbon dioxide is required inside the cell and or a large amount of oxygen needs to move out of the cell leaf, at that time stomata remain open. So now we come to from where different substances are obtained. Now, as is clear to you, carbon dioxide is obtained from the atmosphere, mainly in case of land plants. But where water plants are there, aquatic plants are there, then how will they obtain their share of carbon dioxide? In case of aquatic plants, carbon dioxide gets dissolved in the water and moves inside the plants, leaves of the plant through simple diffusion. Water and minerals are absorbed by the roots of land plants, and in case of aquatic plants, directly water supplies the minerals and nutrients to the plant. The energy required for the process of photosynthesis is obtained directly from sunlight. Is sunlight the only source of photosynthesis to occur? The answer is no. We can carry out photosynthesis even under artificial light, and this process has already started being utilized in some countries, especially US, where a single large container is converted into a vertical garden, and therein light is given artificially to the plants for their growth. Heterotrophic nutrition we will take up tomorrow, so we are stopping here. You can also join me on my Facebook page and Instagram account, the links of which are provided in the description box. Thank you.